So first up, let me have the privilege to welcome you all in our today's webinar. This is the second webinar and commercialization. To briefly talk about his background, he completed his Bachelor of Science in Mechanical Engineering from Bangladesh University of Engineering and Technology. And then he completed his Master's from University of Akron and his PhD from Case Western Reserve University, both in mechanical engineering. He has authored multiple research articles, and currently he is working as a staff engineer in the research and development department of Goodyear Tire and Rubber Company. Today we are lucky to have him among us, as he's going to talk about the research perspective from an industrial perspective, because he's working in an industrial environment. So it will be obviously an excellent opportunity for us to hear from him. So being that said, I think we can start our today's webinar. I'd like to request Dr. Muhammad Noman Hassan to kindly take over from here. And after the presentation, we can take the questions from the audience. Dr. Muhammad Noman Hassan, if you please.
Thank you, Shourav. Thank you very much for the kind introduction. Welcome everyone to my talk. I believe most of the audience, the time zone is, it's night there, and some of the audience is here in the US. So good morning and good evening to everyone. So the topic of my discussion today is the miniaturization of medical device. So this is something I worked on in my doctorate study, so I'll talk about that, but I'll not only focus on just the research side. I'll try to provide different aspects, a lot of aspects along with the research aspect that can be I hope that can be helpful for a lot of the youngsters here. So before going into the discussion, I'd like to briefly talk about myself. So Shourav actually told a lot of it here, so I'll just briefly discuss about my background. So I have a sort of diverse background, I would say, because I mostly worked on the application side for computational fluid dynamics, numerical heat transfer. I also work on experimental sides for microfluidics, additive manufacturing, manufacturing process modeling. I also work on like rapid prototyping and product development, even during my PhD. So I believe having experimental and computational background gave me a bigger perspective on what I'm doing. And I believe that's something everyone should consider if someone is going to decide that I'm going to work on like computational side or experimental side. Just my personal experience or opinion. So I'm going to talk about sickle cell disease, and I'm going to talk about a point of care technology to detect and identify sickle cell disease. So here's some background for that. So we actually designed, developed, and validated a microchip electrophoresis system. And we're going to talk about how we developed it, what are the components, and we also developed the test procedure. So we'll talk about those things as detailed as possible on a higher level. Then we'll talk about some of the characteristics of the system because that will give us a better idea to understand the system. And then we'll talk about the validation, like real life application and the commercialization. So what is hemoglobinopathy? So hemoglobinopathy is, in simple words, it's a hemoglobin disorder. If someone has a defective or hemoglobin or hemoglobin disorder, that's what's called hemoglobinopathy. So the normal hemoglobin is called hemoglobin A, and there are like more than 200 variations out there. So here's a small list of most prevalent hemoglobin variants you can see here. And we're going to mostly focus on hemoglobin S and then C and E. So we know like how the genetics works. For every characteristics, there are two genes, right? So for our blood cell, we can have, if you have a normal hemoglobin, then we'll have a hemoglobin AA. Then based on what kind of variants someone has, there can be like homozygous variants like hemoglobin SS that you can see here, or like heterozygous like hemoglobin S. So when someone has hemoglobin SS, that's called sickle cell disease. And when someone has a combination of A and S, that's called trait, because that's not a complete disease, but the person can share some trait or show some trait of the disease. So it's actually a very prevalent disease. And in terms of like the whole hemoglobinopathy, like around 7% of the whole world's population carries some sort of or some variation of hemoglobin disorder. So it's not a small number. And if you just look at the sickle cell statistics, it's like millions of people have the trait, and thousands of people, hundreds of thousands of people have the disease. And the disease has like severe consequences. So just briefly to discuss about sickle cell disease, the sickle cell hemoglobinopathy can be categorized like this. So the normal is the normal or controlled hemoglobin type that someone have if there's no hemoglobin variants. And based on the combination, you can have hemoglobin SS, that's the disease, just as I said, then trait, and there can be another variation that's called SC. So you have a sickle hemoglobin and then another variation of hemoglobin that's C, and that is also included in sickle cell hemoglobinopathy. So we'll discuss about this more detail down the road when you see the results. So what happens, or what is sickle cell disease? So usually a healthy red blood cell is very flexible and they can go through blood vessels very easily. But when someone has the sickle gene, so it can be either trait or disease. What happens, those sickle hemoglobins can get easily deformed and stiffened. So they actually blocks the blood vessels, and that can cause a lot of health issues, and they can be severe, and they can be as severe as like mortality. So this is a very significant health issue that you cannot overlook. So just look at the statistics. There are like hundreds of people, children that that's born in the Africa, they have sickle cell disease, and not to include the trait. And almost 90% of them will not survive to their adulthood. They will sometimes they die before even 5 years old. And the global prevalence can be even higher in like South Asia or like Mediterranean region. And it is expected to grow because as the population grows and there are more migration and interracial marriages, these kind of issues are not globally bound anymore. They actually spread pretty quickly. So you can see it's a global concern that's growing. So let's look at the sickle cell disease and the economic aspect of it. So on the top, you see the global prevalence of hemoglobin disease is highest in the African region. Then we have some in the South Asia, and in American continent, we have some, but not as severe as Africa. But if you look at the GDP and the PPP of around the world, you'll see the region where you have the highest prevalence of the sickle cell disease, they have the lowest GDP, and they have the lowest economic capability to address this healthcare issue. So it's an irony and it's a concern. You can see here that this region has the highest prevalence, and this is like one of the lowest GDP countries. So if you look at sickle cell disease from a first world countries point of view and a developing or underdeveloped countries point of view, we're comparing the life expectancy of the citizens of United States. And that's for like 100 years. You can see all Americans who doesn't have sickle cell disease, they have a high life expectancy compared to the people who have sickle cell disease. But in Africa, you see it's even worse. But in USA, things started to get better when the government started to take this seriously and took some measures. And those measures took a lot of money. So because of the economic might, US was able to pull this off, but in resource limited countries, it's not possible actually. So that's a concern. So what happens in the developing countries and underdeveloped countries? You have lack of technology and you have lack of trained personnel. And what it causes, that treatable disease that can be addressed, people who can be helped, they actually go undiagnosed. And those can result in early death. And there's an awareness about it, like all the statistics show that if you look here, the huge number of children born with this hemoglobin disorder and the life expectancy, which for person with sickle cell disease in Africa compared to the other countries, even in Jamaica, it's much higher than Africa, than US, UK, it's very high. So we see like there is a disparity in healthcare for first world country and the underdeveloped and developing countries. And why is that? We're going to talk about that as well. So the question is, can we provide, we engineers, can we provide any solution to that to address this disparity? Because everyone has a right to have a healthy life, right? So we're going to ask a series of questions, and the answer will answer our previous question, like, can we do something? So the first thing, or the first question is like, is there an awareness or unmet need? Like, do we really need to do that? If people believe that. If no, then there is no point in working the solution. First, we have to create the awareness. But if we have an awareness, next question is, if we can come up with a solution, is this clinically acceptable or credible? Because we engineers think like engineers, and doctors and clinicians, they have their concerns. So there has to be a synergy. So if the solution is clinically credible or not, if not, then we have to come up with something that's credible. But if yes, then the question is, can we actually integrate it to the current practice or current healthcare system? Because we just saw like there's a big difference how healthcare works in the first world countries and the resource limited countries. So it's very important that we do not need to develop any additional infrastructure to implement those solutions. If it's not possible to implement, then the whole point is actually pointless, because even if you have a low-cost solution and the solution is clinically accepted, but because of the cost, you cannot implement it. But if it's possible to integrate in the current practice, the next concern is sustainable commercialization. And if we can do that, then we have a potential point of care solution to address this kind of health issues. So what is point of care solution? I'm going to briefly explain. So we all have seen like quick test for diabetes and pregnancies. You don't have to go to the doctors. You can actually buy the tool and test it yourself. So those kind of devices are called point of care testing. So what point of care testing does, takes the diagnosis capability from lab or doctors and put it in the general people's hands so that more people can test themselves. So that increases the number of people that can be diagnosed and screened and get accurate and better healthcare. So now that you know why we need that and what do we need, let's look at like what we have developed and how we developed it and how it corresponds to those questions we just asked. So in this slide, you can see some of the current options that we have available for detecting sickle cell disease. Here we have hemoglobin electrophoresis and isoelectric focusing. Then we have HPLC. This is slightly complicated than, that's somewhat complicated than hemoglobin electrophoresis or IEF. And it's more costly as well. And then we have gene-based testing that's even more complicated and even more expensive, requires sophisticated facilities. So what happens, almost all of these tests are lab-based, and usually they're centralized lab-based. You collect sample from the nation, you transfer them to some centralized lab, they do the testing and come back with the results. And that increases cost. There is a burden of having a vast infrastructure. You need trained personnel in the lab. It takes a lot of time to get the result. They have a lot of installation cost. So most of the things doesn't apply to those countries that you just saw, like where the disease is most prevalent. So to address that, people are working to come up with point of care testing that can give you results in a couple of minutes. And we have an example of point of care testing here. So what we have done, so we actually thought of starting from the lowest low cost solution and can we improve that. So in the previous slides, we have seen that hemoglobin electrophoresis is one of the lowest cost technique. We were thinking, can we make it even lower cost and portable and make it a point of care system? So if you look at this cellulose acetate based hemoglobin electrophoresis, we see it can detect all the variations, hemoglobin variation or hemoglobin variants that's associated with sickle cell disease, right? AFSC. So it shows that it clearly has the capability to detect the sickle hemoglobin, so hemoglobins associated with sickle cell disease or sickle cell hemoglobinopathy. So what we have done, we actually have made an automated system that takes a sample, runs with a control, and then gives you a result. So what's different from the existing technology? Or what's the novelty of the technology? So it's miniaturized and portable. It's like a small suitcase. You can just carry it in your hand. The lab-based system is pretty big, and it takes a lot of, it has a big footprint compared to what we have developed. Also, the benchtop system or the standard system has a control. You have to run your sample and a control. And there has to be a trained person who can interpret those results compared to the control and give you the results. And the traditional system is just qualitative assessment. It just gives you an idea what kind of hemoglobin you have. It cannot actually quantify, do not know what's the percentage of each types of hemoglobin you have. So what we've developed, it can show a quantitative results as well. So we'll see down the road. So we talked about the clinical credibility. So the solution we have developed, is it clinically credible? So WHO Health Organization actually recognizes sickle cell disease as a global healthcare problem, and they actually provided a list of tests that is acceptable for wide level screening, and hemoglobin electrophoresis is one of them. So just from that, we can say that hemoglobin electrophoresis is a clinically acceptable solution. And if we can make it low cost, it will be accepted to the clinical community. So how does it work? So in a layman way, we actually take a blood sample, it can be like a one drop or like a few drops of sample, and we just mix it with a standard calibrator marker. We run the test. This is standard hemoglobin electrophoresis. We separate the hemoglobin and then we detect. And the whole test can be run within 8 to 10 minutes, depending on the hemoglobin types. And most of the time, it's less than 8 minutes, actually. So we talked about like, we're going to talk about the cartridge technology, readers and software. So the whole HemChip technology has a cartridge that runs the test. There is a reader who actually runs the test inside the cartridge, and there's a software that controls everything. So we're looking at the cartridge now. So this is the cellulose acetate strip where the hemoglobin gets separated, and we have all these components to facilitate the test. These are the electrodes to apply the external voltage, and we have some in-chip blotting system that controls the weightness of the paper when you run the test. So once we have the samples loaded and buffers loaded in the test, we can run the test, and that separates the hemoglobin, and then we identify and quantify the hemoglobin types. So this is the first prototype that we developed of the reader. So it guides the user to run the test. It runs and does a real-time image acquisition and analysis, and then gives the results with the help of this software. So this is also like one of the earliest version of the software that we developed. These are much sophisticated now. So Hemex Health is a company, it's a startup company who actually worked with us to commercialize this technology. We'll talk about Hemex's contribution in detail in the later section. But most of the development on the hardware and software side, the Hemex Health has like majority of the contributions. We provided the technical details, and they actually made it happen. So this industry and academic collaboration actually really like made it possible to make this technology. So what do we have here? We have a test that is really low cost. It's $2 or less. It runs the test in 10 minutes, where the traditional test can take 30 minutes to an hour. It's mobile, it's robust. And the way the test is designed, it can be run with anyone with minimal training. Like if you just read the manual, you can do it. And why, we'll talk about this later. So now we have a solution that is low cost, clinically acceptable, and integrable to the system because we don't need big infrastructure to implement that. All we need is like a sustainable commercialization. So in a nutshell, we see that we are dynamically tracking the hemoglobin as it gets separated. Then during the test, we look at multiple time steps and we detect the hemoglobins. And once the hemoglobin is detected, we do some image analysis to enhance our quantification. And at the end of the results, you can see what percentage of what hemoglobin you have. So that has a lot of clinical relevance. So if you can detect the types of hemoglobin and if you can detect the percentage of it, that actually dictates the therapy that the patient needs. So having that quantitative result or quantitative capability really gives an edge to this technology. So we actually faced a lot of design challenges when we were developing this. One of the major challenges, the in-chip evaporation and condensation. The reason we decided to talk about it because it will dictate the later development of the technology. But you can see we actually rely on optical imaging of the hemoglobin separation and image analysis to give the result. If you are not able to look at the bands of separation, it's really difficult. So we actually come up with, we tested a lot of options, but we come up with a very low-cost solution to do that. You can see the difference, like on the top you have the condensation and the bands are very blurry. If you look at the red line, you cannot tell the difference, like which band is where. But if you look at the bottom image, you can clearly see separate peaks that can clearly tell the difference between two different hemoglobin types. So we saw the cartridge before, but that's not how the cartridge started. So initially, we actually used to make the cartridge by layering up a lot of polymethyl methacrylate, PMMA, to make those chips. Those are very laborious and time-intensive processes. But once we came up with a design that can be mass manufactured by injection molding, we were really able to run a lot of tests and the process become really simple. It was possible to do that because we had a grant that allowed us to do the initial prototyping and the mass production. And we worked with a local company that's called Thogas Products. They really helped us to come up with a modified design and to make it mass producible. So here you can see like another synergy between academia and industry and how it can influence the type of research and the quality of the research you can do. So we talked about how the cartridge works. We'll talk about now how the test is done. So when you want to run a test or you want to test a patient, we take a cartridge and we have to go through some preparation. So that's what is shown here. Then we collect the blood and we do some processing of the blood sample as well. And then we have a special applicator that we use to transfer the sample to the cartridge. So I'm going to briefly talk about this. So this is like a controlled capillary device that allows us to transfer the sample here. And we also have some additional techniques that can help us maintain the wetness of the hemoglobin separation medium that's very crucial for the test. So this is a demonstration of how the test is done. We've seen this before. I'm just going to quickly skim through that. So here's a quick video. I believe this will help a lot to understand the whole process. So I'll just show the video. I don't know why the video is not running. If not, I'm just going to skip that for now. So this is one of the important slides that I want to focus. So when we are developing the technology, we had a lot of failures. And we actually looked into the failures to make the system robust the way it is now. So this is a Pareto chart where we actually plotted the types of error with the frequency, and we looked at the cumulative percentages. That actually gives us an idea, like where should we prioritize for improving the system to make the system robust. So to briefly tell you, like we actually categorize all these errors in 3 different categories. The categories can be process error can come from user interaction, and that can be, and the third one is a combination of both of these two. So based on this, we actually come up with a pretty elaborative algorithm that's embedded in the software that we talked about that guides the user from the cartridge preparation through the sample preparation and running the test and getting the results. That actually makes sure that even a non-trained person can run the test. And we actually talked about it, how we made sure that a minimally trained person can run the test. So I'm going to quickly talk about the detection method, how it works. So we say like there are 2 different types of hemoglobins that can be homozygous or heterozygous. For homozygous, you have only 1 band. For heterozygous, you have 2. So when you run the test, we looked at the band, we detect the band, and based on some threshold, we identify the band. Sometimes it's possible to confidently identify the band, then we just show results. If not, we do a retest. So this is an example of homozygous sample. We look at hemoglobin A and hemoglobin S, and we are clearly able to detect and identify the hemoglobin bands. If we have heterozygous band, we have multiple bands, then we look at all the bands, and then we apply some threshold. So based on the threshold, if we can identify that bands easily, then you don't need to do a logical correction, then you just publish the result. If we have some difficulty, then we actually apply an additional, or like a set of additional logical correction that gives us the band identification, and then you publish the result. So there are 2 examples of that. So it actually seems like it's easier for heterozygous sample to detect the band more easily, and then homozygous band, because for homozygous band, we actually don't have any inconclusive test. So we have been looking at this video. So what it does, we actually dynamically track the hemoglobin mobility, and there are a number of metrics that we look into, and based on those metrics, we can actually detect what band, which band is what, and we can do it pretty accurately. So here's a number of tests we've run in the lab setting. Those are like done by the trained personnel, and you see like the accuracy is really high. This is the distribution of the bands for homozygous sample. This is the distribution of the bands for the heterozygous sample. And we see we only had 1 sample that we say it's incorrect, it's actually incorrect. We had to go for logical correction. So because that was a heterozygous sample, we were able to apply the logical correction, and even though the band was identified at the wrong region, we were able to call it correctly. So that's how the logical correction works for this technology. So that's what I just mentioned. For homozygous sample, it's much easier to confidently provide the result. We also did additional statistical analysis. So the Pearson correlation is pretty straightforward. We compare our result with a clinical gold standard, that's HPLC. So the Pearson correlation actually shows our heme chip result is linearly correlated with HPLC. And what the bland output analysis does, it compares 2 different technologies. So here we plot the mean. For the same sample, we actually take the mean of the heme chip result and HPLC, and here we plot the differences. And based on these analyses, the position of the mean and the distribution along the mean actually tells us if there is a systematic bias on detection, because if you look carefully, you see that hemoglobin A's are more on the higher part of the mean, hemoglobin A are more on the lower part of the mean. So that says like the technology you've developed has some systematic bias for specific hemoglobin types. That helps us to like calibrate our system to provide better quantitative analysis. So I'm going to quickly go over the thermal characteristics just to give an idea what kind of characterization we have done. So once we run hemoglobin separation, we apply some voltage on a buffer that has a sample in it. 2 things happen. Because of the nature of the buffer, electrophoresis buffer, we have resistive heating, and we also have electrokinetic flow. So the electrokinetic flow actually governs the follow-up electrolyte flow, and the resistive heating actually contributes to the evaporative loss. So those two are kind of interconnected, and they actually have a cyclic nature. Both of these components actually further influences the electrokinetic flow. What happens for the evaporative loss? We have runtime pH change of the separation media, and we have change in humidity inside the cartridge that actually causes the condensation that we saw previously. So this is a very complicated interaction of physics. If you look here, we have electrolyte transport, we have chemical changes happening, we have heat and moisture transfer. So even from experimental point of view, this is very complicated. To look at a specific aspect of it, it's very difficult. And if you think about a computational point of view, it's even harder because if you just look at the separation media, you have a two-phase electrolyte and vapor because electrolyte is getting evaporated. And if you're looking at the whole system, when you include the cartridge, the empty space inside it, you also deal with the air that's present there. So you have a combination or a mixture of gas, and you have two-phase flow, and the system becomes really complicated. So I'm just quickly go over how we did the pH tracking. So we actually developed an image analysis system, the hardware, the software, the image analysis code. So what it does, it actually looks at the separation media, the presence of some standard color, and we captured the images. And these actually helps us later to come up with a calibration curve. I will talk about the calibration curve later. So to actually make this image analysis possible, we have to do uncompressed image capture. I can talk about this detail, but it's just itself can take hours. So I'm just going to quickly go over like what we do. We actually do uncompressed image analysis, and we actually save the images as a planar format to make it more efficient for further analysis. So it shows how this is how the image is stored in the imaging system, and this is the process we do to still come up with a planar image system, and then we apply our image analysis code. So this is the calibration curve we're talking about. We actually developed some solution at different pH. We imaged them in the presence of that color standard, and it came up with color for each pH. That gave us this calibration curve. So what do we do with this calibration curve? So we talked about this image acquisition system just like 2 slides before. That actually helps us to run the test. And we talked about the calibration data, how we came up with the calibration data just in the previous slide. So based on that, we run a test. We do some test preparation, we run the process, we image it, we encode it, and store it. So once the test is done, we go back, then we apply our image analysis algorithm. And based on the calibration data, we can actually show the pH change in the cellulose acetate paper. So this is one of the results that you can look into. You see the pH on the paper at the beginning of the test, how it changes, and what happens at the end of the test. And you can clearly see here the pH shift or pH change on this axis with time. It actually starts flat, then we have a large pH change, and then becomes stable again. So we actually ran this test for different runtime, different buffer, and different test parameter, like different voltages as well. So this is like a sample test data that we're looking here. Next, we'll look into the heat transfer aspect of it, because that's very important for multiple reasons. So here you can see we actually developed another calibration system that helped us to come up with accurate measurement of the temperature change on the paper. So we actually have a thermal imaging system. We heat the paper at specific temperatures and we image the paper. So the software that comes with the imaging system actually give you some temperature reading, but this reading depends on a lot of factors. The emissivity of the surface, the ambient temperature, the calibration of the camera. So just looking at the temperature here is not going to give you the accurate result. And that's what happens most of the time, because the low-cost IR images, IR cameras are not properly calibrated all the time. So based on these setup, we actually came up with a calibration curve, we see like, even though we see some variation, it's pretty much linear. So we actually had a very good system of infrared imaging. Then we apply this calibration to the runtime images you see here, and we come up with these thermal distribution plots. So all these plots are different points along the road. We are looking from the cathode side to the anode side, and you see like there is a dynamic change in temperature, and temperature change at different point actually varies with time. So we have spatial temperature difference. We have temporal temperature difference here. So the most interesting result we saw here, like if you see the earlier stage of the test, the hotspot actually at the middle of the test, but as the test progresses, it actually shifts to the anode side, and that actually affects the hemoglobin mobility. Because if you have increased temperature, you can see the temperature goes to like 60, 70 degree. That actually denatures the hemoglobin, and that can affect how the hemoglobin moves. So both from system point of view and from clinical point of view, it's very important to have this understanding and calibrate our system to get a better result. So this thermal mapping actually gave us a lot of insight to optimize our system. I'm going to quickly talk about some of the computational work that we have done, but it's a very simplistic work. We actually looked into the nature of the evaporation from the cellulose acetate paper to match with our experience that you see when you run the test, and actually matches pretty easily, but pretty quickly. But this is not an easy simulation, and we simplified the process a lot. So no way it's a complete representation of the system, but at least it provides some idea how the evaporation actually changes the temperature along the paper and how the wetness varies of the paper. If you're interested, I can provide additional resources that can help you to understand how the modeling is done. So I'm going to quickly go over the validation and commercialization. So we actually went to different parts of the world and worked with local people, local agencies to implement this technology. Here we see, like in Africa, Nigeria, there are already established vaccination programs. What we did, we worked with those NGOs and local governments to include our testing with the existing technology and existing infrastructure to have an additional test done for sickle cell disease. And all we had to do is like add a new line in the existing form. So it's pretty straightforward, and we don't need any additional infrastructure to do that. Here you see we very minimally trained person running the test in real world setting without us showing them how to do it. And here we present the test results from 3 different parts of the world. All of these tests are done by local people with minimal training without us interfering or showing them how to do it. And he came up with a very good number of tests that gives us very good results. We have a very small number of tests that where we were not able to make conclusive results or reliable results. So for the commercialization, I mentioned before, we worked with Hemex Health and a lot of other companies to make this product commercialized. This is the first product that we developed, and we worked with a lot of third-party companies in collaboration with Hemex Health to actually get the final product that we have, and that's much more sophisticated than what we had before. And here is a slide we see like where we are running different field tests now, current field tests and future tests that we have planned. So the technology also got regulatory clearance from a lot of the part of the world. We already got clearance in the Europe, so, and also in India. So we can actually start distributing the technology there to be implemented in the real world. So the doctors and clinicians can implement that for a diagnosis of the sickle cell disease. So here are some of the awards and recognition. The technology has been recognized as an honorable mention for the Patents for Humanity by the US government. And we have received a number of regulatory approval to implement this in real world. This is another thing that we have been working on. This is making a smart infrastructure for the testing where the results can reach directly to the doctor and the patient via mobile technology. So this is another work we have done, and actually won an award in this competition, and we have been funded by this agency from NIH. So just quickly, you can see how the project progresses. Here's the list of milestones, how we started, what we did, when we licensed, when the technology was licensed, when we started the technology, where it is. And here's the list of funding agencies that made this work possible. So I'm at the end of my slides. I'd really like to acknowledge the guidance that my advisor provided me. He gave me complete freedom to lead the project, and that actually helped me to improve me as a researcher. And not to mention all the countless people. I cannot list everyone here, all the people who helped me down the road. And it was a real collaborative project. Here's just a small number of people that I can mention, and all the additional funding agencies. So that's all of my presentation. So if you have any question, I'll take questions now.
Thank you very much, Dr. Mohammad Nomar Hassan, for presenting in front of us. This was really a nice example of research being commercialized and that being used in real life applications. So we'd like to take questions from the audience. Feel free to hop in, or even you can type in the chat if you have any questions. Okay. So I see a question from Khan Mohammad Rabbi Bhai. Should I read it loud?
Sure, please.
Yeah, he's asking that how does the non-condensable gas impact the performance of the system?
So Rabbi, thank you for the question. If you can elaborate the question a little bit, that will help me to answer the question. So if you please.
I think Rabbi can unmute himself and ask the question.
Yeah.
So thanks, Naman Bhai, for your great talk. I was just wondering, since basically it's a evaporator, and so it's a pressure-driven system, right? So from your evaporator, you basically create heat, and it evaporates out due to the heat and moisture transport. You have a change in capillary pressure. But I was wondering, and you have the condensation side as well. So I was wondering, in your liquid side, if you have any non-condensable gases, such like NCGs, that does not really like to be condensed. So will it impact on the performance of the system? That's what I'm wondering about.
Okay, that's actually a very good question. So we actually explored a lot of aspects of the condensation and evaporation. The first thing we try to do is like, can we stop it? The answer is no. I mean, by nature, these buffers produce a evaporation and condensation. So the buffer, those are already clinically established for electrophoresis, does not have the capability to actually stop the evaporation, not that I know of. So then we actually looked into, if we cannot stop it, can we live with it? So later the focus actually shifted on, can we mitigate the address of imaging, not address the evaporation, at least at the earlier phase when we were just developing the cartridge readers, because most of the work was just on the design side, not on the pure science side at that point. And also I'd like to add that when you have a commercialization aspect, you have to make your system like really repeatable. And there's a cost involved to that. So what we did, instead of like developing or trying different buffer, we actually did some design and manufacturing changes that helped us to cope up with the evaporation and condensation that we see. But in short, if I like to answer your question, I don't know how that non-condensable gas would have impacted, because we didn't explore that. But if we can stop the evaporation, that will definitely impact the temperature on the cellulose acetate paper and the pH. So what the evaporation does actually cools the cellulose acetate paper. If we don't allow the evaporation, the paper will get even hotter. So you've seen, like, we start from a room temperature, the test. I'll quickly go back to that slide. We start from a room temperature and the temperature goes to like 60, 70 degree centigrade. At this level, the hemoglobin already gets denatured and its nature changes, its mobility changes. If we don't allow the evaporation, the temperature will go even higher, right? It will even degrade the hemoglobin more. So in a sense, if you think that the evaporation is a blessing in disguise, but that caused us the imaging problem. So we decided to take care of the imaging problem, not eliminating or minimizing the evaporation. Does it answer your question?
It in fact did, Norway. Thanks for this response. So I actually have some other questions regarding, so what program did you use for your thermal mapping? Is it a ResearchIR program?
You're talking about the software or the device?
Yeah.
Okay, we actually, please go ahead, finish your question.
Yeah, so I was wondering, how did you ensure that you have emissivity of 0.9 of the paper? Because that definitely is very sensitive in measuring temperatures using infrared cameras.
Exactly, so that's what we talked about here. So this is a reading that we get if we just analyze the result here, right?
Yes.
So we actually didn't look into this result at all. Like, we didn't take this for granted. So what we did here, we actually heated the cellulosic paper at different temperature, the temperature you see here, right? And then we took thermal images of the heated paper. Then we matched the result that we got from our IR camera and IR software and the temperature reading that was showing in the temperature control device, right? So that helps us to build this calibration system. So what we did later, we did not actually just took the IR result for granted. We took the result and we recalibrated. Shouldn't say recalibrated. We actually normalized those results based on this calibration curve that made sure that the results you're looking at is not dependent on the emissivity. While saying that, we tried our best to maintain the same setup that we used for the calibration system, because if we had even if we change the illumination slightly, it will impact the results greatly, right? So you have to take the results with some grain of salt, because we were not super sensitive about keeping the illumination consistent, but we tried our best. All I can say, like, the temperature profile you see, that will remain the same even if it impacts the result. So even if you don't want to take the absolute value of the temperature plot with absolute certainty, but you can actually look at the trend, how it goes, right? So that itself gives you a lot of insight how the system is working. Does it make sense?
Yeah, it does make sense, Manwai, yes. So I was kind of expecting that as well. So thanks for your response. Sorry for taking all the time in the Q&A section. I have two more questions. So
okay.
Another question, Manwai, is in your initial designs, you have so many layers. So I was wondering, how did you make sure that you did not have, you have very good sealing in your system, and how you bonded each layers?
Okay, let me quickly go to that slide. I believe it was here, right? So we actually use a very thin layer of double-sided adhesive. And once you apply that adhesive and apply the pressure on the whole assembly, it actually remains air and watertight up to some pressure. But if you look at the application, we actually don't apply any strain on the assembly that could make the seal inactive or like start to peel off, right? We don't apply any pressure, we don't apply any tension. So once it's sealed, it remains pretty much airtight and watertight. When I'm saying airtight, I'm talking about the joints, not the hole that we have on above. So that actually worked pretty well, but the process is really, really time consuming and laborious.
Yeah, thanks for the response, Noman Bhai. I have one last question, that is,
sure.
How did you, in your PhD journey, so I'm currently a PhD candidate at UCF. So I was wondering, what advice do you have for current PhD students and also future PhD students to get US patents and get their doctoral research out from their lab? So what advice and tips do you have?
Okay, that's actually, I would say, like one of the best questions I have ever faced in the talks, because sometimes we are so much into the scientific aspect of our work, we actually forget that it's also important. We need to have our research reach to the general people, right? We need to have a technical outreach. Unfortunately, it's not just the student's responsibility. It depends on the lab culture. It depends on the PI. So not all the PIs will be willing to go for commercialization. I was lucky that my PI is very much interested because he works with healthcare related issues. He likes to make those technologies available to the people as soon as possible. That allowed me to have this opportunity. But this is not the standard practice. But that's not the end of the world. You can always talk and try to convince your PI to actually do that, at least to go for the patents, right? So you yourself cannot just apply for a patent. Your advisor have to approve it. The school have to approve it. They're like technology transfer office who looks into your claims, looks into everything, and then they go for the patent. And you have to spend some money to maintain that patent. So I would say if you really want it, talk to your PI, try to make a compelling argument that is worthwhile doing it, and good luck.
Okay, thanks for the responses, Naman Bhai. Thanks again.
You're most welcome, Rabbi.
Thank you, Rabbi, for your questions. Yeah, these were the question I think some people also asked in the chat, and also I had this question. I mean, I think someone asked that if you please share your journey, how you got to work with. So I think you partly answered that question.
Mhm.
And so to emphasize on that question again, so I mean, what are the challenges did you face during the patent? I mean, of this device while you were doing the patent?
So if I understood the question correctly, so are you trying to understand how the patent is different from like a regular publication, or how do we do it? But I'll try to answer the question as best as possible. So publishing patent is not much different than publishing a paper. What I would say is they're just different in nature. How you write it is different than how you write a paper. So one major difference I would say, when you write a paper, you're writing for a scientific community. The language you use, that's different from the language you use for a patent, because when you're writing a patent, you're writing it for the legal community. Lawyers write and read the patents, right? So I would say if you really want to understand how the patent works, I would say like read a lot of patents if you can, because they're really, really long. But most of the time, a lawyer will do it for you. But to know how patents are written will actually help you to actually focus some aspect of your research. Because sometimes for publication, you focus on some things. In legal claim, actually focus on something else. So I'd say like get idea how the patents work or what kind of claims you can make. So if you know what kind of claims in the patent can be made, that will dictate your design of experiments and help you to come up with acceptable patent more efficiently.
Okay, yeah, thank you. So yeah, there was a question from audience to please share your journey, how you got to work with this and how you prepared the design initiate.
So when talking about the journey, are you talking about my journey up until my PhD, or like the journey of my PhD for this project? If you can clarify.
Yeah, I think she mentioned as a whole, I think, yeah. This
Okay,
was
so
her question. Yeah, share your journey, how to go, how you got to work with this? I mean, maybe this product development, medical device.
Okay, I got it. So in our lab, when I was doing my PhD, so no one actually works on a single project. So this is actually not just the only project I worked on. Actually, this is the third major project I worked on. I actually worked on some other projects as well. So we actually started this project as an undergrad design uh, course. Like some undergrad came to us, they want to work in our lab and come up with some design project for their course. So it was really just to help an undergrad to come up with a design idea for his design course. But once we started working on that, we actually saw the potential here. So then it became a master's project for another student. His name is Ryan Ung. He did some clinical work before me. I was still working on that project when Ryan was working on that. So when Ryan left, we actually saw some potential because we presented those results in some conferences, and people was actually interested to see how widely and how effectively it can be applied. So we were thinking about, at that point, we started thinking about this project seriously, and we started spending more time on that, but still it was not my main project, even though it ended up being my PhD thesis. So about like a couple of years later, when we started to receive some funding for that, and Himex personally started to communicate with us and showed the interest about this, then actually we started putting some patent for that. And at some point, the project became so big, it could not remain like a side project. So then I took up this project as my main project and started focusing on this primarily. Then that's how I just got into the project, and to get the things done. It has been a long and wild ride, I would say, in a positive way. I actually got to work with a lot other companies, not just the companies I mentioned here. I worked with a company called Andrews Cooper, who actually developed the reader for us. Focus product actually came up with this design that you're looking here. From this one, we actually worked with the engineer, expert engineers, because the design has to be had to be transferred in some way so that it can be injection molded. And then we worked with a lot of contractors who were really expert on the software side, image analysis side. So we were doing the research part. We came up with the things that we need, but we could not do or need more expertise. And we kind of gave that work to other companies or contractor, and then worked with them to develop it, test it, validate it, and then implement it in the result. So at every step of the way, we had to go through this cycle. Look for people who can do this that we cannot do, explain them what we need, work with them, come up with the initial prototype or draft, test it. So when you test it, it doesn't work the first draft. So keep working on them, keep finding errors. So we actually showed a slide where you look into the failure mode or failure analysis, right? I believe it's somewhere down the road here. So this is what we did at every step. This is kind of like a grand summary, but we did in every step for the cartridge development, for the reader development, for the software development. We prioritize what we have to solve now, what we can solve later. That's how things started moving. And it really took me at least 2 years to have a working system to start getting the data that I can publish. So it was a long wait. My PhD actually lasted for 6 years. So once we had the system, we started working on the limited number of samples that we had access in the US. We came up with some validation of the system. Then we went to Africa, Thailand, India. We trained people and started getting those data. And to build up that capability took us like at least a year, because when you take a system from the lab, give it to people who doesn't have any expert skills, things can get messed up pretty easily. And you can see in this plot, like it really got messed up. And then we had to come up with like a foolproof algorithm strategy, how we can transfer this technology to like anyone who doesn't have any science background or like technical background. Once we established that, then we started to get our data and those field tests. So when we got a very good understanding of the system, very good established method, process, then the commercialization actually took flight. And once we knew that our system is working, then we started focusing more on the basic science, like the characterization part. I actually quickly went over that, but we actually did a lot of characterization. I just provided some sample of what we have done. So that's how the project ended up being so vast, I'd say. Does it answer your question?
Yeah, I think that was really a good story and a lot to learn from the story. I think the present students will be greatly benefited, and also current PhD students like us.
Mm-hmm.
So that there was another question from the audience. Is this device inspired from any other previous model?
When you say previous model, if I understood the question correctly, there was no previous point of care testing based on electrophoresis. There are a lot of point of care devices for different diseases, and it's a $1 billion industry. So the idea of miniaturization of medical device is nothing new. What we did, the novelty of our system is like we took a very low cost system that's not considered as a high reliable system. We made it reliable, portable, and rapid. So we shouldn't take claim that we actually developed the electrophoresis system. We actually made it a point of care system that is reliable and rapid. Does that answer the question?
Yeah, I think, yeah, it helped. So I think we are almost out of time and we should call it a day. If anyone has any more question, you can.
Yeah, I'd like to jump. Thanks, Shiro. Yeah, I think it's been an hour. Hi, Nuanvai. This is Vivek.
Hi, Vivek. How are you?
Yeah, how's it going? Yeah, it's a nice, interesting presentation. I mean, pretty applied to real life. I mean, not everyone gets to work with this kind of project, so it's interesting to see something coming into real life, becoming all these things. So yeah, I don't have any questions. Like I understand biology very surface level, so I don't get there. But I want to point out two things, that some of the things that you mentioned, like Pareto charts, and then you have got like statistical analysis. These are things very important, and generally engineering students are not taught well somehow in undergrads or grads. Like somehow surface level teaching has been done. So I would really like, how did you like, because like people don't understand, people always underestimate the value of statistics and all this kind of like failure analysis in their studying and then research as well. So do you have any suggestion on that for current students?
Yes, thank you very much for the question. I actually have a very strong feeling about statistics that's not taught in good undergrad mechanical level, but it should. And I also have a strong opinion about how it's taught. And I have seen people actually abuse and use it to actually, you can actually bend statistics to prove your point, because statistics is not mostly like making a hypothesis and proving that hypothesis. The way someone devises the hypothesis can actually take the focus from important aspect to a non-important aspect. So I would say it is something that should be included in undergrad level as a like must, like the way we learn math, calculus, this kind of math. I actually was invited for a podcast where he talked about just statistics and how it can help and how it can mislead researchers. So I can actually provide two very good articles. If you're interested, I can send you the articles or link to those articles. I would say it's a must read for anyone who's dealing with statistics for their research, because one of the paper actually shows that almost half of the literary article that you can find online where people use statistics are used in a wrong way. And that's mostly because they don't know what that actually means. So when you implement statistics, it's very important to understand what we do. And most of the time people do like ad hoc, like they do the work first and then try to come up with a hypothesis that justifies what they did, and then they do the statistical analysis. But it's acceptable to some extent, but not as a sole method. Like ad hoc statistical analysis is something you do to like provide additional basis to your primary hypothesis. So statistics should be dealt when you're designing your experiment, come up with your hypothesis, and then run your experiment, and then try to prove your hypothesis, not to come up with a hypothesis that matches with your result and then running the experiment, because it will always match.
Yeah, that's true.
If you devise your statistical hypothesis after seeing the results, you know what to expect then, and you don't know what you know what worked at that point. So you can take it to your advantage and come up with a hypothesis that's already it will be proven. So I'd say like that's most of the people do it because it's not taught to us like when we should look into the statistical analysis. And I'm no different from that. Like I actually learned that a long time after I started working on those things, but at least I learned, I feel happy that I at least know that I didn't know that.
Yeah,
but
that's true. I mean,
to conclude the answer, like if you want those articles, I'd be happy to provide those. And I'd say those are very valuable resources for any researcher.
Definitely. I'll send you an email, then I can share with other people as well. Yeah, that's a nice answer. And I will always like, not everyone is going to do modeling or like computational aspects of the research, but if you're doing some sort of like testing and experimental characterization, statistics probably something that you should learn. Even as an engineer, you should always learn. Yes. Even if you're doing modeling, you probably look into experimental data and should be able to understand what's going on.
Yeah. So if you're actually working on pure science, you're coming up with some empirical rule, you have to know statistics, right? Because then you're just looking into data and like trying to make a sense out of it.
Thank you for your answer.
You're most welcome.
Okay, anyone else have any question for Noman Bhai? Okay, I think there is no more question. It was really a nice presentation, and we got to learn lots of real life perspective of product design and how to get your research outside of the lab. I think this will really help us and give us some practical perspective of research and development. So with this, I think we can end our today's webinar at this point. Thank you very much, Noman Bhai, for presenting your work in front of us. We are wishing you all the best with your patent and all. Please share those articles with us. If Vivekda can share those with us, if you share those with Vivekda. And thank you very much, everyone, for joining the webinar. So we will meet again in the next month, hopefully. Take care. And Noman Bhai, if you have some last, words, yeah, to share with us.
Sure. First of all, like it was my pleasure to present my work here. It will give some meaning to my work if it motivates anyone to do anything in future. So that's all we can aspire, right? To motivate the future generation to do even greater work that we could not achieve in our lifetime. So I would say, don't lose hope, keep working, and good luck.
Yeah, thank you, Noman Bhai.